this video, I'm going to show you how to conduct a dihybrid cross. And when Mendel coined the term dihybrid, what he meant was that there were two traits being considered. So in this case, the traits are being denoted with the letter A and the letter B. And you want to make sure that if you are reading a genetic story problem and you determine that there's two traits, that you select unique letters for each trait. And the hybrid part comes from the fact that this parent is big A, little a, big B, little b. And this diagram is showing us the arrangement of these two genes on separate homologous chromosomes. And I wanted to show you this diagram because it's showing us all the different ways this parent could generate gametes. So if the homologous chromosomes lined up like this in metaphase 1, it would be possible for the big A and it would be possible for alleles to have a big A and a big B. If they lined up like this parent would also make gametes that have the little a and little b allele. It's also possible for the chromosomes to line up like this in metaphase 1, and in that case, gametes would include the big A, little b allele or the little a, big B allele. This is called the independent assortment of chromosomes, and this is where we get the law of independent assortment from the fact that the chromosomes could line up either this way or this way in metaphase 1 and produce all four of these allele combinations in the gametes. So now let's look at a genetics Punnett square for a dihybrid cross. So in this case, we're looking at characteristics in P's, and I wrote out our grid. We have big R denoting the roundness of the P's. We have little r being recessive to big R, and if you have two copies, the P's will be wrinkled. We also have big Y being dominant and conferring yellow P color. If an individual has two of the recessive alleles, they're going to end up with green P color. So we got to think back to meiosis and write the different gametes on the two axes of this square. So assuming independent assortment of chromosomes, this parent could make eggs that have any of these allele combinations: big R, big Y, big R, little R, big Y, and little r, little y. And we just looked at that. That's because of the segregation of alleles into gametes and also the independent assortment of chromosomes in meiosis. The other parent has the same genotype. They're also heterozygous for both of these traits, so they're going to be able to make the same gametes: big R, big Y, big R, little r, big Y, and then little r, little y. So we do the cross the same as we did the monohybrid cross in each of these squares. We're showing ourselves the genotype, and we can figure out the phenotype if we use our little map over here of each of the possible offspring of these parents. So what I want you to do now is pause the video and continue filling out this square. So these take a while. You got 16 squares. You got to be careful and make sure you don't miss anything. Hopefully you took the time to fill out your square. it should look something like this. And now the last step in any genetics cross is to figure out the ratio of phenotypes present. So I see 1, 2, 3, 4, 5, 6, 7, 8, 9 individuals who are who have round yellow seeds. I see 1, 2, 3 individuals who have round green seeds. I see 1, 2, 3 individuals who have wrinkled yellow seeds, and I see 1 individual who has wrinkled and green seeds. So this is what Mendel consistently found, this 9:3 to 3:1 ratio, when he conducted dihybrid crosses from true breeding parents. Thanks for watching.